Biophysics applies the theories and methods of physics to understanding how biological systems work. Biophysics is essential in looking at how the molecules of life are made, how cells move, and how the complex systems in our bodies function. Understanding how all life works requires experts from a wide variety of fields, and it takes a vibrant interdisciplinary community to make that happen. Now, in San Diego, California, scientists from all over the world are coming together to share, learn, and discuss the very latest in biophysics. This is the 2020 Biophysical Society Annual Meeting, and we're Biophysical Society TV. Well, we are here in San Diego, where experts in everything from chemistry to physics to engineering and much more are coming together in one place. And you know it means one thing. It is the 2020 Biophysical Society Annual Meeting. With lectures, symposia, poster presentations, social events, and much more. This is the very place to be for the very latest in the dynamic field of biophysics. And we are here to cover it all. Welcome to Biophysical Society TV, bringing you updates on everything Biophysical Society related, plus previews of this week's coming events, and a very special look at the exciting research happening all over the world. You can watch right here at the Convention Center, in your hotel room at the Hilton or Omni, and of course on the meeting website or app. Now let's see what's on the show today.
I think biophysical research right now is at this sort of renaissance point. New tools that enable us to go inside living cells and make measurements directly there on either single molecules or small populations of molecules, molecules in complex mixtures. And so all of that's really well represented in the meeting.
We have a growing population of female members. We went from 10% up to 35%, and we would like to see that 50-50. Our students are 50-50.
What I really enjoy and what I think is great about these types of workshops at the Biophysical Society meetings is it allows you to come and sit down for 20 minutes or a half an hour and really learn something new about a new area of research that's at the cutting edge of biophysics in 2020.
To get us started here at the Biophysical Society Annual Meeting and to give us an insight into what to expect over the coming days, we are joined by two hardworking meeting co-chairs. Hello, Patricia Clark and Bill Coberts. Thank you so much for joining us. The first question, either of you can answer, what's gone into getting this year's meeting off the ground?
This has been a 2-year process, developing the program, putting this together. Over that time, Bill and I have had a number of conversations and drawn heavily from the member-suggested input for design of the sessions.
Yeah, I mean, Patricia and I have been coming to this meeting for 20 years as, graduate students. We had never met until 2 years ago when we were signed to be part of this meeting. And the two of us now have really taken those 18 years of experience of being attendees and trying to put together the best meeting that we can.
And it's great to know that the two of you met 2 years ago here.
Yeah.
So how big is this one this year?
So my understanding is we're expecting about 6,500 attendees, 101 invited speakers, and in addition to that, an even larger number of platform short talk speakers, and many, many hundreds of posters as well.
Is there anything new this year?
There's a lot that's new. I think that's part of the excitement part about the Biophysical Society meeting is that as co-chairs we can bring in what's new and exciting. And I think one of the things that I think we're particularly excited about is the workshops where we've had these are Tuesday night meetings, I believe, and they focus on specific biophysical tools that people are developing to help spread them out to the society at large. And we've had a great time putting those together, and each one of those topics is quite new of the four of them.
Well, you answered my next question, which is what are you most excited about over the next few days?
I think biophysical research right now is at this sort of renaissance point, right? It has a long history of experiments in test tubes at equilibrium or with very purified components. And so there's been a real kind of renaissance towards new tools that enable us to go inside living cells and make measurements directly there on either single molecules or small populations of molecules, molecules in complex mixtures. And so all of that's really well represented in the meeting.
Any advice for attendees?
Get the app because there's so many concurrent sessions that you really want to be able to kind of go through the program information and search out the topics that are most of interest to you and make sure that you can kind of program your time.
In addition, if young investigators, there's a lot of career development workshops that are not part of the scientific component, but learning how to become a better scientist and get to where you want to be.
So there's a whole parallel program basically alongside the scientific program. There's a whole career development program, and so that's definitely something especially younger first-time attendees should be aware of. I want to make sure that there's a special plug for a session that's going on tomorrow evening, Sunday evening, which is the society has really decided to take a leadership role with regard to sexual harassment at annual meetings. This is something that scientific societies as a whole are starting to try to explicitly address. And the Biophysical Society has decided to take a leadership role in this and actually have an explicit session at the meeting for a conversation to start about sexual harassment at the meeting. We don't really know how much of that is occurring, but given the size of the society, there's got to be at least some, and we want to actually be out in front of that and providing resources for people and support for people to know who to report to and so forth.
And lastly, I think the other thing that's really the best part, I think, especially as a young person coming in, is going to the poster sessions where all of the advertisers and all the companies are there with all the latest and greatest equipment, and it's really it's just a lot of fun. Plus you get a free t-shirt or two, being a fan of t-shirts.
Yeah, I'll have to remember to get that on my way out. It's going to be a great meeting either way. Thank you so much for joining us.
Thank you.
For having us.
Well, it sounds like it's going to be a great meeting. We'll be hearing from the meeting co-chairs again a bit later on to hear a rundown of today's most exciting events. Stay tuned for that. But first, Biophysical Society TV showcases some of the most exciting research happening around the world in biophysics. And coming up next, we'll head to the Center for Cellular and Biomolecular Machines at University of California, Merced, to hear how they're looking at everything from biosensors to circadian rhythm. But first, let's take a look at what else is in store.
We are interested in biofilm formation. To study bacterial adhesion, we use modern surface science techniques such as photoelectron spectroscopy or atomic force microscopy.
Our center research focuses on the macromolecules and machines of life. This goes from nanoscale assemblies all the way to cells and how they organize to form colonies and differentiating tissue.
For us, we welcome new users, and that way we bring all the community users together and we together discuss about their research leads.
As scientists, we have to make knowledge available to the broader scientific community. This is really at the core of what university research is supposed to do.
The Center for Cellular and Biomolecular Machines is an interdisciplinary center that brings together more than 20 faculty labs from across physics, bioengineering, material science and engineering, chemistry and chemical biology, and even molecular and cell biology to study how biological entities come together to achieve function. UC Merced is a new institution that was established about 15 years ago, and it's been growing, and it was established without any departmental boundaries. And even now, even though we have grown a lot, that still is part of our basic DNA.
Our center research focuses on the macromolecules and machines of life. This goes from the nanoscale assemblies that are composed of proteins to all the way to cells and how they organize to form colonies and differentiating tissue, and including also interactions with synthetic materials. So we do all this type of research in an interdisciplinary and integrative way, and then we work all together towards these goals.
We work on the circadian clock system of cyanobacteria, and the reason why we work on it is because we can reconstitute the entire clock in a test tube in the absence of any living cells, which means that we can look at how the clock proteins interact at the molecular level without any complicating factors that are associated with live cells.
So one of the applications that the center is really looking forward is try to develop biosensors that will be used for diagnosis and for biomedical research. One of the key problems that biomedical research has nowadays is that it's very difficult to monitor the different processes at the cellular level. So by developing these biosensors, which are based on proteins, we should be able to probe different properties in the cells, including pH, the concentration of different molecules inside, that are key to understand all these phenomena.
My lab is all about taking optical engineering technologies and applying them to biomedical problems. So we build new microscopes to try to answer biomedical questions that have been unanswered because there's no technology to do it. So we build advanced microscopy for intravital imaging. We also do intravital sensing of things like oxygen in tissue at very small volumes. And we're looking to branch out into other techniques as well.
My research is on using nanomaterials to put them together to make sort of novel multifunctional hybrid devices. So being part of CCBM actually got me started on this collaboration. So we make these nano-assembled shell structures that we can enclose things in and break them with light or heat. And that's the sort of the platform that my group designed. But collaborating with them, we are actually translating it to work in tissue engineering platforms. This is where the collaborative part comes in, and it actually lets me get a new and really exciting way of applying what my group with other physicists have sort of come up with.
We provide a very rigorous graduate program that combines coursework and hands-on training modules that are required for all our graduate fellows. And so the goal of this program is to actually provide every student that comes through our center with the basic biophysical toolkit that allows them to be versatile. And this is a critical skill for cutting-edge research in biophysical sciences, biotechnology, and biomaterials, and it also makes them very attractive to the biotech industry.
The center really focuses on collaboration, and I don't think that there's a career out there anymore where you are not going to be collaborating. So it's a skill in learning how to communicate your research to people outside of your specific area, how to communicate the data that you're acquiring to other people, and it's just what you need in order to move on.
It's been really interesting seeing the different work that's been going on in the community, all the different things about protein folding and tumor research. It's all going towards the hard problems that we face in society. And so that's been very fascinating to see what's been going on and seeing how I can contribute to that as well with my research.
Me being a computational biophysics student, I tend to interact and collaborate with experimentalists regularly. So it's one-of-a-kind training experience.
UC Merced was chosen to be here in Merced because there is a great need for more economic development, a research base, which is what UC Merced brings to this area. As an NSF project, our center contributes to broadening participation efforts. What that means is we are committed to increasing the representation of women, students with disabilities, and various racial and ethnic groups into STEM fields. Our center initiated the Science and Technology Enrichment Program, which is a program for high school students from the area. This program allows students to come in and gain research experience in faculty labs. They also engage in a one-week orientation at the beginning of summer. They attend scientific talks throughout the summer, and then in the fall they come back in our open house to do a poster session. We're proud of the fact that through our center and the funding and resources it brings to UC Merced, we're able to do new things that were not able to be done before this came. So we're excited about the new opportunities that this center provides for young people in the area, not just the undergraduates and the graduate students, but those who are living in the community.
So my vision for the future of CCBM is to be an institution on campus that is a focal point for all biophysical science, biotechnology, biomaterials research, and also an entity that provides graduate education and training, including graduate minor degrees in these fields for all students from across the university.
Exciting work there from California's own UC Merced. And as we just saw, training is so important for this often technically demanding field. And to discuss how the Biophysical Society is providing workshops to do just that, I'm joined by Professor Henry Colcraft. And Professor Colcraft, what's the name of the workshop you're working in? What's in it?
Well, it's called Chemical Biology Tools for Biophysics. And we have 5 speakers in there who will be talking about various aspects of chemical biology and protein engineering as applied to molecular and cellular biophysics and protein structure function.
And who should come to this session?
Everyone. This is a general talk or general discussions that will be useful for everybody interested in molecular and cellular biophysics as it relates to protein structure function. So I think it will be very interesting for protein biophysicists because they can learn new tools to apply to their own questions.
And just to reiterate, why is a workshop like this so useful? Why should people come out?
Well, for every field, it's very important to get new tools and new strategies to answer questions that otherwise would be impenetrable. And this sort of workshop is very important for people to come in, learn about new things that they can apply to their own proteins of interest and ask questions in ways that they probably haven't thought of before.
All right, well, thank you so much, Professor Colcraft, for your time. We've also got a preview of some of the speakers that's coming up. Let's take a look.
The subject of my workshop is going to be applying chemical biology to a family of voltage-gated ion channels. And what I would mean by that is we would like to use this method as a means to encode new types of chemistries into ion channel proteins. What's particularly exciting about the work that I'm going to talk about is it involves a number of skill sets that cover a broad range of modern molecular biology and chemistry. So we're going to be talking about organic synthesis and the making of new types of amino acids and how we try to encode them genetically into ion channel proteins. So attendees will learn about chemistry, chemical biology, and electrophysiology.
Stefan Pless from the University of Copenhagen, he'll be talking about protein splicing elements known as split inteins and how these can be utilized to introduce synthetic peptides into proteins.
We're talking about some technology we've been developing to kind of spy on spider toxins as they interact with proteins, voltage-gated ion channels in the membrane, to see what the channels themselves are doing through the eyes of a spider. I hope people get the kind of mechanistic idea of what we're working with, as well as a really good idea of the limitations of trying to look at protein conformational change in this way, to inspire but kind of ground in reality in terms of what you can get out of this type of technique.
Kimberly Beatty from Oregon Health Sciences University will be talking about using genetically encoded tags for fluorescence and electron microscopy. So in every field, new tools or new techniques is very essential to ask new questions and to gain new insights in ways that they haven't thought of before and advance the field.
What I really enjoy and what I think is great about these types of workshops at the Biophysical Society meetings is it allows you to come and sit down for 20 minutes or a half an hour and really learn something new about a new area of research that's at the cutting edge of biophysics in 2020.
You can get a more almost informal, really, as I say, grounded discussion of what it takes to use these kind of techniques and how far you can push them and how far you can't.
So please come to our workshop Tuesday night, 7:30 to 9:30 on chemical biology tools for biophysics.
Make sure to check out those workshops on Tuesday evening. Now to another group facilitating the future of research. It's all about open science and open hardware in Lausanne, Switzerland. Let's go to EPFL's laboratory for bio and nano instrumentation, where they're preparing for the future of academic research.
I'm a first-year PhD student at the Laboratory of Bio and Nano Instrumentation, and I develop microscopes to look at biological samples with high resolution. My goal at the beginning was to build an instrument that I needed for my own research. And then afterwards, when the instrument was built, some of my colleagues became interested in the project, and we are in an interdisciplinary institute, so it was very easy for me to talk to colleagues in biology, and then we started sharing the research. And progressively it becomes something more open, more shared, and we could share the instrument in several labs.
Open science is going to be the future of academic research. That has many reasons, and funding agencies all over the world are starting to demand that all research results that are obtained are being made freely available to the scientific community and the general public all over the world.
This is a way that shortcuts some of the delays involved in going through a typical commercialization route and the loss of technology during that long process, and it enriches the scientific community. It's a very direct way of translating research into changes in the community that matter.
As part of what we do in our research, we develop new measurement tools and primarily also instruments. And there was a big barrier between labs adopting our tools, because for them they were big black boxes, they were a bit scary, and they definitely could not build them by themselves. So what we decided to do is we organize workshops here at EPFL where we invite people from other universities to come and participate.
When people come for a workshop, they receive all the individual parts for assembling one of our microscope heads. After this assembly period, they will go to the lab and test and learn how to use the microscope on their own.
When you build a microscope to share for others, you need to take into account that they will not know the system. So you need to build something that is easy to use, that is properly documented. And in this case, because it is open hardware, we need something that one can build without using too complex instruments, that does not require too many tools or too heavy maintenance.
We have 3 different types of workshop. They all are part of this AFM technology, but they involve different components of the system. The main workshop that we host is a workshop based on the atomic force microscope head. This is essentially the heart of the instrument. Another one is we do workshops building very small range and very high-speed scanners. And lastly, we do workshops on the controller. This is actually working on the electronics and on the software to actually control these instruments.
I came to the Open High-Speed AFM workshop here a few years ago. Over the course of the 2 days we did a lot of testing, so you put something together and you test it. And that was really excellent for us because it means we can take things back to the lab and we can know if something goes wrong, we've got a bit of an idea how to start troubleshooting them ourselves. And any changes we've wanted to make or anything we've needed to do with them, Georg and his students have all been really communicative about how best they would do it or their approaches or sending us details of suppliers. So for me it was a really immersive and professional experience that left me going away knowing exactly how the thing that I'd just made worked.
I think for me the most interesting part of this workshop is actually to meet people from all over the world, people who are working using the same instrument, but obviously the applications are so diverse, and it's great to learn what they're doing, and we can get ideas from them and they can get ideas from us.
It is a solid foundation for both collaborations and getting citations of your own work.
We are planning in a large network grant now across Switzerland to incorporate more universities and more researchers in this general idea and extend it to other kinds of instruments than we've used already, and even see if these kind of ideas might work for other kinds of technology.
In addition, we feel that this is part of our job anyway, because we're not just being paid to develop technology and develop new knowledge, but as scientists, we have to make that knowledge available to the broader scientific community and the general public. And so this is really at the core of what university research is supposed to do, enabling new discoveries and doing that in an efficient manner.
Now back to the meeting co-chairs for a rundown of Sunday's unmissable events. Take it away, guys.
Today is Sunday, the first day of the symposia for the conference. That includes symposia on fuzzy interactions and crowding, which really speaks to the in vivo environment and the movement of biophysics to in vivo experiments. And also single molecule studies, trying to see not just ensemble behavior, but individual molecules, in this case, for this symposium, specifically with regard to transcription, translation, and splicing.
So my Sunday favorites are going to be the mitochondrial section on how calcium gets in and out of these organelles in the cell. It's actually a pretty debated topic, and there's this thing called the mitochondrial permeation pore, and there should be some pretty good fireworks that I'm looking forward to. And the second topic that I'm excited about for Sunday is the session on ion channels, or anion channels, and actually that's incorrect, because there's actually going to be some anion transporters in there called CLC transporters, and the new anion channel on the block, the TMEMs, which are really exciting, and I'm looking forward to hearing what Chris Hartzell has to say about his work on that session.
Coming up, we're going to be having an in-depth discussion with Biophysical Society President Dr. David Piston. First though, we wanted to get a feel for what's hot in biophysics this year, so we went to the experts, you.
Yeah, at this moment, to me, super resolution microscopy is a really exciting area, because even just few years back, the Nobel Prize has been awarded in this field, and now still the field is growing, and also due to the super resolutions, it is being applied in the biological science, and it's really exciting to me.
I'm into optics, so I really like building and using lasers to try and analyze different biological components, and so I think that's the big side is optics for sure. Personally, I really like single molecule techniques, so like optical traps and optical tweezers, and angular traps and that kind of a thing, yeah. That's what I like.
Yeah, so I think that the structural membrane biophysics, so some of the structural work that is being done to understand the structure of some membrane proteins using things like cryo-EM is very exciting. Also, the machine learning tools that are allowing us to analyze data in ways that we didn't understand before help us understand new physics is also very exciting.
I would say the liquid-liquid phase separation of proteins, and then also liquid-liquid phase separation in lipids.
So it's the area of Titan research. I'm interested in the role of Titan in regulating active force. So it has been shown that Titan has a big role in passive force, but we are now interested in its role in active force production in skeletal muscle.
The Biophysical Society plays a crucial role in bringing together scientists from all over the world, and now taking a lead in how that all happens, it's the Biophysical Society President, David Piston. Thank you so much for joining us. First of all, what's big in biophysics this year, and what do you expect will get most attention this year?
Yeah, predicting what's going to get the most attention at the meeting is always hard. There's so many diverse things that are happening. But some of the things that are exciting me before seeing the presentations is a big focus on biophysics of health and breaking biophysics out of molecules and going into organisms, even into human bodies. There's stuff on cardiac, on heart conditions, stuff on Alzheimer's and aging and the biophysics of the molecules that play a role in that. And this is, I think, emblematic of how biophysics has evolved over time, from really something in a test tube to something that really is happening in affecting human health. And I think that's still the growth area. There's still a lot of new techniques being discovered all the time and developed, and that the membership of the society is really the people that take the lead in that. And so I'm excited to see what the latest and greatest toys are on the exhibit floor, what the latest and greatest new developments in instrumentation there are in the poster sessions and in the talks, and also how those are being applied then to human health or plant science or the whole range of biology.
And what's the last year been like for you? And really, what successes and challenges have you seen in your leadership role?
Yeah, well, being the president of any organization is usually a be careful what you wish for kind of proposition. Over the last year, we've spent a lot of time trying to increase transparency of the organization. A lot of people who have been members for many years didn't know how certain parts of it worked. How does the program come together? How does council make decisions? So we tried to really up the transparency, posting a lot of information about the council meetings, putting more timelines on everything, and that's been good. There's been obviously across the world, the Me Too movement, and that's affected STEM just as much as anywhere else, and we've been trying to confront that. We have a symposium at this meeting about that, which was developed over the last year, new code of conduct, new reporting software. So those things have all sort of, everything takes a little bit more time than you think. I think they're all good ideas, and trying to get that all done has really been taking up my time.
Well, okay, good to know. And where do you see biophysics making the biggest impact going forward? And what research areas can it really make breakthroughs in?
Yeah, so I think that the one area where you're going to see breakthroughs coming right away is going to be sort of drug discovery, where the biophysics is defining how drugs interact with the target molecules, how to find the target molecules, how those are distributed inside of the body. Certainly when you go outside of human health and you get to plant science, any sort of things that are genetically modified, how can you make those safer? How can you make them better? How can you make them faster? And the biophysics and the biophysical techniques are really affecting all of those things. I think that's still a big growth area. What we're trying to do at the meeting is capture the wealth of information that's coming in, feeding back into what new techniques do we need in order to solve those important problems.
How are you working to ensure the society works for everyone? And why is that important?
Yeah, well, certainly if we only have old white males like myself in the society, our membership will be declining. Science is growing as fields, diverse people from all over the world, as diversity across the world. The Biophysical Society, 30% of our members are international, not from the United States, and we want to grow that. We have a growing population of female members, as we went from 10% up to 35%, and we would like to see that 50-50. Our students are 50-50. We have more students of color, and we need to have encourage them. If we do not encourage them to stay in the field and make these contributions, they're going to be lost forever. And we cannot rely on only part of the talent to solve the problems that we have in this society, whether it's the problems of population growth and how that's taxing the world. Biophysics can affect a lot of those things, but I don't know the ideas, but the young people do. And that is important to basically cast that net as wide as possible.
Well, great. And as you work towards that, what is next for you?
Well, next for me is, of course, I have to be past president for a year, and that will involve nominating the next slate, working on nominating the next slate of officers. And after that, I think I might take some time off and actually be able to see all the science that happens at the meeting rather than spend a lot of time working in meeting rooms and in conferences.
Excellent. Thank you so much for joining us. David Piston with the Biophysical Society. Thank you.
Thank you.
Great to see how the Biophysical Society is growing and benefiting its members. Now we've come to the end of our first show, but it is just the beginning for us here in San Diego. Tomorrow we'll hear from a couple of this week's symposia speakers on kinetic stability and sensation membrane proteins. Plus, we find out what lies in store for the future of biophysics lecture. Until then, make sure to watch online, in your hotel, and around the convention center. We'll see you tomorrow.